Excellent presentations, and now's the time for some discussion. Yeah, you were first up.
Because I have something specific for Amy. So, Amy, so I have a tankless water heater in my house. Does that mean I'm not at risk, or I just have a whole new set of risk?
Excellent question, and I did not plant that one. We are working on that. So we have a grant from NSF where we've installed full-scale water heaters, including a tankless one. And so our hypothesis is, and since we're on a roll with hypotheses, is that you are at less risk. But there's a lot more tubing in there, so there's more surface area. And they don't always heat up great. I don't know, maybe you've had a good experience, but.
Not really. I mean, overall it's okay, but it takes we're wasting a lot of water because it can take minutes to get up to hot water in parts of our house.
Yeah, this is not so much, Amy, this is directed towards you and your colleagues at Virginia Tech. And this is not so much a question as an observation. I find that there's very little to take comfort in the whole experience in Flint. But I will say that watching your colleagues being interviewed and in congressional testimony and so on, I've been very impressed by the professionalism of that crew. And I want to say especially the students. I've seen some of the students who are out in the field making measurements talk to the press. And I got to say, I'm really just blown away by their professionalism. I think that must speak volumes about the quality of your educational system. So I just want to compliment you. And I hope you'll take that back to the team at Virginia Tech. You're doing a great job. Thank you.
Thank you. I'll definitely take that back. And I kind of realized that about the students, too. At first, I was a little worried about, ooh, they're talking to the press. And then I realized what a great job they were doing and just kind of let that happen.
Did you train them before, or do they have innate gifts?
Well, I'm trying to be humble here. No, they really are amazing students. But we did have a team in place. And there are, we do emphasize teamwork. And two courses that I'd like to think were influential is that Mark teaches an ethics class, and he's very hands-on with that. I mean, he really takes that class seriously. And I teach a team science class where students learn about working in interdisciplinary teams and some of the ins and outs of that. And so I'd like to think that that played a role, but thank you.
Sharon, question?
Amy, I have a quick question for you. So I know Maria Domingos Bello has looked at Amerindian tribes and seen that they already have antibiotic resistance genes, even though they've never been exposed to any kinds of antibiotics at all, and are not drinking water from any of these systems. So it seems to me that antibiotic resistance genes are just in the environment, and we're constantly exposed to them. So I was just wondering if you had any thoughts on how your research dovetails with some of what we're learning about the natural unexposed environment. Because
That's a very good question, and for sure there are natural background levels of resistance. Most of the antibiotics that we use are derived from natural compounds, and I think Gloria's work is an excellent demonstration that even in human populations, and people have done work on wildlife populations, that there are antibiotic resistance genes there. But what type, what diversity, where are they coming from? Are they associated with mobile transfer elements? Are they really scary ones like the New Delhi metallobeta-lactamase 1 gene that encodes resistance to just about everything? Which hosts are they in? So I think, as was discussed in some of the other presentations, we need an understanding of what is the background, and then what are humans doing to contribute to a change in that background, and is that change a cause for concern?
I have a sort of group question. We, ended up with a discussion of plumbing, and the earlier ones focused on the air, and how do we make this link with bioaerosols and jumping from one to the other?
I'll just take the simplest stab in that, and we know microbes partition both directions, right? And I think the shower illustration that Amy gave exemplifies that. We know in the natural environment, microbes partition in both directions. So I think we've got some basis with what to make that, to make the jump that you were just suggesting. Is it understudied? You bet. And I think that we're going to have to be careful to define that in terms of exposure risk, what we know about that and what we don't. But both, I think, have to be a priority, Joe. We have to look at both and continue to look at both.
Amy, I'd like that idea that you kind of put forward, a core microbiome of tap water, I guess it was. Could you tell me what are a few common members of the tap water core microbiome? And what are the source? Is the source the premise plumbing? Is it the distribution system? Are these microorganisms that may have already been disinfected? I'm talking maybe about the ones that don't change significantly. The common members, not the rare members.
That's a good question. I'd have to look up exact numbers, but when we showed the beta diversity plots that were the normalized, then there was quite a bit of overlap. So the majority are pretty similar. There's kind of just a municipal water type signature. I'd throw 90% out there, just ballpark, that are pretty similar in the core microbiome. But I might double check that number and get back to you.
I'm asking.
Where are they coming from?
Well, just what are they? What genus do they typically? If you said what's a core microbiome of indoor air, I might say staph and strep and Carini bacteria.
There's obviously a lot of proteobacteria, a lot of actinobacteria, and then there's some odd ones like the what you would think are photosynthetic bacteria, but I don't know where they would be getting light, so they might be carrying through from reservoirs, or it might be that we just don't have a full understanding of some of the cousins of these organisms and what they're actually doing in water systems.
And I'm just to give Hal his due here. He was joining remotely, as you all know, and he actually had just emailed me a comment on kind of related to this point, that there was a paper, I'm guessing recently, in an environmental studies and toxicology journal about airborne antibiotic-resistant bacteria, geographically located near farming. So that's maybe another source we can look at. So we'll have to get Hal here in person to tell us more about that, but I wanted to share that.
Shoot.
So just a comment from, the perspective that I was sharing in the talk, is that all the talk about antibiotic-resistant bacteria, particularly when it's pulled from metagenomics, is a little tricky, because if it were as big a problem in some sense as we would like to say, the treatment failure rates should be higher uniformly across the board. But they're not. In some kinds of infections, it's a really serious problem. And in other kinds of infections, treatment failures aren't very common. And so, there's a big debate in the Forum on Microbial Threats type crowd, which is different than this crowd. Treading carefully into the ID territory is probably wise.
Dr. Haas.
Yeah, I'd like to pose a question for everybody, and that is, If the objective is to assess impacts on human health, is genetically based information enough? Are the viability measurements sufficient to overcome those limitations, or do we need something else?
I could probably say, much like with Jill Banfield's work and some of Amy's work here, I think we used biomarkers, and they were important, but we really need to really understand the total genomics, I think, is the next step. You know, we can go beyond that. What are the proteins expressed? But I think knowing just what are the genes expressed is the next step in the progression, because just knowing that an organism is present, you could say that Francisella tularensis was found in the air. Francisella tularensis has an infectious dosage response of 1 to 10. So if you're seeing it in the air, why aren't people getting sick? It's because it's not really the pathogen strain. It's close environmental variants. So I think we really need to know much more detail than most current studies do. It's the current studies. It's, you know, at this point we can only see very distinct genomic information for simple communities on the order of tens to hundreds to maybe a few hundred, whereas many environments are thousands to hundreds of thousands to millions. So we have a ways to go yet, but I think that's going to be the next important step. But the second part of your question was, I think we have to be a little careful here, because I think for this field to grow, human health has to play a role, just because that's where the money is. But I think we want to, what's really important, and maybe this is just from a scientific standpoint, is I think we really need to understand some of the basic sciences behind it. So I don't want to put the cart before the horse, so to speak, and I think that we still need to understand. I think even though health is important, this program got started just looking at the basic microbial ecology, and I think that's still a very important issue.
I'd like to throw in on the microbial diversity problem. Evgeny Sokorenko was looking at E. coli ST131, and that's a sequence type. So each strain is identical at 8 of the common housekeeping genes to the base pair, not even pointless changes, if you say. And a good number of them aren't pathogenic. And meanwhile, so then he had to look at other genes that weren't housekeeping genes, and they start to have explanatory power as to what attached where and in the human body, and you started to break things out into UTI causing and gastrointestinal causing and so on. But the fact that they could be identical at 8 housekeeping genes suggests such a high rate of homologous recombination amongst these organisms that they're seeing each other, and yet they're still maintaining their ecological status. So the amount of differentiation that we're going to need to do is really intense. When I pull a panel of 100 Acinetobacter from clinical wounds and I run them against each other to see which ones kill each other, they all almost kill at least one other member, and they do it in hundreds of different patterns, and they're all coming from wounds, and many of them are the same sequence types. So they're behaving radically differently. And we haven't even begun to scratch the surface. So the moment that we say putative pathogen based on a genus, we're already wrong. We need to be so careful about that, because it'll mess up all our risk understandings. Meanwhile, the cart and the horse with the health, if we're not actually measuring real health, then forget the cart. We've lost that entirely.
So I also think that in our work and many other people's work, we see it's not, things don't have to be viable to cause health effects in the microbiology world.
Yeah, actually, it seems that most current studies focus on diversity. So looks at like literature published. I think you raised a very good question. We really should move beyond diversity. So most important activities. So if we compare different habitats like the water and the air, I guess the air, the microbe is not active there. So in the water, maybe biofilm is much more active. So really we need to find out what's active. So, but I don't know, is maybe for these speakers, is there any good tools we can look at activity more efficiently, more effectively? So rather than just look at the diversity. So I think the field need to move beyond the diversity.
I can just briefly answer for me. So we look at RNA as a proxy for activity. It's you can go through a whole list of why that's not good, but there's not much better in our for simple work in our case, and so we still feel that under certain specified cases. Acknowledging it's not perfect, and there's many examples to show to the contrary, that we can use RNA expression as a level of activity. And so just even RNA concentration, difference comparing RNA to DNA ratios is what we do sometimes.
Yeah, so another question just Benjamin mentioned, so also I think that we have to move beyond the 16S. Because so far, most of the studies for microbiomes, instead of shotgun sequence, you focus on the 16S. So 16, because the limitation, conservation limitation, you say you get the genus resolutions. So you talk about the differences among different genuses. Just Benjamin mentioned even E. coli 157 is different strains. So we have to move to the function gene marker rather than the financial gene markers. So I know the function marker much more difficult if you do shotgun sequencing. There are a lot of artifacts that are very difficult to data analysis. But in order to get the deep understanding, maybe we have to think about the function gene marker rather than the financial gene markers. Yeah, it's useful, but I think this is rough level.
I was going to say I completely agree with everything you said, Joe. The unique thing about the indoor environment is that you're concentration limited, and that horrible step of PCR for doing 16S is really a lifesaver for the indoor environment, because you can take DNA that you can't measure and you can amplify it and you can sequence it and you can get diversity in some information. If you want to go with RNA, you're going to then not be able to handle air samples, or in many cases surface samples as well. So I completely agree. We would need to find ways to understand function much better, and all of that stuff, transcriptomics, microscopy stains. PMA is probably the best thing out there right now to do it. But the indoor environment has some unique aspects that the human microbiome and wastewater treatment and many other well-studied environments, the soil as well, don't have limitations in.
So then you say the one need could be, you see, so we should beyond just analyze the samples, use molecular tools. We should think about what kind of technology we can improve the sampling. So we can do large sampling, not more sampling, more biomass. So how to manipulate the samples. So I know this most people are ecologists, but I think this is a very critical part if we want to do things correctly.
A generic question for the group. Several people have brought up the notion of a core microbiome that we should be trying to identify. Is that even a realistic goal?
I don't know that I have the answer to that, but I know that we struggled and debated what is a core microbiome. So there's somewhat of a standard definition that it's an OTU that occurs in all of your samples or 99% of your samples, but is that really the best definition? So I guess just broadening your question a bit further. So
just adding to that, I don't think there's a core microbiome. I think it does vary. I think it seems like what some of the studies are showing is that it's the complexity of the community. So in other words, it's microbial diversity that's important. As you go, no matter if it's in a human health or in indoor air related to asthma, it seems like the more diverse communities seem to be more beneficial, and the less diverse, less beneficial. So I don't know if there's a single core. It's just there are some, maybe there's some core benign type of organisms that in more diverse communities. But as you go down in less diversity, you seem to find a higher concentration of pathogens.
Yes, Jelena.
Yeah, I would follow up because that was one of the questions or suggestions that I did have. Rather than identifying all-inclusive or final definition of whether there is a core microbiome, I think there is a usefulness in being able to compare maybe one, maybe two, maybe three. I don't even know. The number doesn't matter, but there should be some cross-referencing between the studies on the one that seemed to be present. For example, for office spaces, I picked one that we did find in all of the office spaces. So I suspect that from the base study. So I suspect that homes or other indoor spaces would have at least one or two or three reference ones that do not have necessarily to be pathogens or sources of disease, but serve as a baseline to kind of benchmark each lab's approach to either measuring, methods of measuring, methods of creating a database. So if we have a common 1, 2, 3, 10, 25, we can have a common way of reporting data without constraining your freedom of exploring. We can have a common method of at least defining how the measurement was done, how uncertainty was measured, and that's really something that can increase the value of the research supported by different agencies.
Yeah, I wanted to follow up. If I understood correctly, the microbial diversity is important for health or for understanding the seriousness of illness? I didn't quite understand that when you were talking about microbial diversity is important.
So it's, I think, a number of epidemiology studies that were done by Finnish groups have shown that having a very complex indoor air microbiome, like what's in a barn or a agricultural setting, seems to be more prophylactic. I was in a study in UCSF on lungs, which so that's more in health and related to ventilator-associated pneumonia and lungs which were colonized. It was found that there was a distinct ratio between those that had lower amounts of diversity, just one or two organisms colonizing their lungs, usually end up being Pseudomonas aeruginosa and a couple other things. Those people more likely died, whereas those that had a higher diversity had a more favorable outcome.
Which would explain a lot of the neonatal results, where the neonatal environments are so sanitized that there's...
Yeah, I think, and I didn't mention this for Jill Banfield's work, but I think that's a real shining example, another, like Flint, Michigan, another example of research that's made a difference on people's lives. Because what this showed was that the indoor environment was colonizing these babies, and it was colonizing them with hospital-acquired pathogens. It doesn't take a Nobel Prize winner to take that next leap. Why don't we colonize them with something else instead? And so I think that work has started. So I think this provided an impetus for instead of having just the random hospital-acquired pathogen affect these effectively sterilely premature infants, colonize them with a healthy cocktail of organisms to create a much greater diversity to make that ecological susceptibility to colonization by these pathogens less likely.
I think Karen was next, and then we'll get down to you, Benjamin. Okay.
Yeah, well, I mean, I'd just like to pull at this before Karen does. The question really, in my mind, is how do our diversity measures interact with our total abundance numbers when we're actually doing relative frequency estimations on limited sampling depths? Because what I see with lungs is that when you get low diversity, it's not actually low diversity, it's high numbers of something that's high on the J-curve, and you introduce something that's going to be high on the J-curve in terms of total abundance, and it's going to push your diversity estimates low because it eats up all your sampling. And I think that actually happens a lot in our relative frequency-based estimations of alpha and beta diversity. And rarefaction doesn't help us much there.
I totally agree. Just to follow up, just that, you know, I oversimplified it. And indeed, that's the case where you have it dominated by one or two. There's still that rarefaction curve, but it's probably the dominance of a few organism that's more important rather than so you can think of it as dominance versus an evenness example.
And what I would think of it is being overgrown by something that just overgrows everything is usually a bad thing.
Yeah.
Okay, Karen.
That wasn't what I was going to say. Jelena, so what we did for the Human Microbiome Project in order to get good baseline points was to develop a mock community of species that was sent to a bunch of labs to get standardized numbers or all that stuff, because I can't imagine a single environment that you can select and call that your baseline for studies in perpetuity. So I think that's something you might want to think about as a community, because that also allowed you to monitor problems in individual labs that were creeping in. And I think the definition I'm hearing of a core microbiome is different from what the rest of the scientific community, the microbiologists use as a core microbiome. So I agree that needs to be teased out more.
Yeah. We have a participant who's in Italy who would like to ask a question. And I know some of you have had your signs up for a while, but let's let poor Hal Levin give a question.
Well, and this is a very specific one to Gary. So Hal says, ask Gary if he knows the contents of a healthy cocktail. Oh. So I'm just sharing that from Hal. So.
I do, but it involves cachaça as a...
Jonathan, you've had your sign up for a while. Go ahead.
Well, actually, it was sort of just, I think it was sort of partially addressed by the group here, but maybe could say a little bit more about your comfort level in terms of this quantification of quantifying abundance levels of a population structure versus just saying whether the organisms are there or not, you know, presence, absence versus relative abundance. And whether you think, you know, the techniques are sort of, sufficient now that you can be fairly comfortable. And I'm thinking more along the lines of whole genome shotgun-based methods in terms of quantifying relative abundance of organisms and what might need to be done there.
Can I heard, first I heard abundance, and then I heard relative abundance. Can you clarify your question?
Yeah, so to clarify, so I'm thinking about how do you characterize whether the abundance of a particular organism is being correctly reflected in the measurement of the whole genome shotgun or other measurement tool in the sampling process, and where we are in terms of state of the art and understanding that.
I could say it's still debatable. I mean, because you mentioned relative abundance and abundance, and that's really important from an ecology standpoint, because oftentimes we measure relative abundance and we're missing the picture if we're measuring log order different units of actual abundance of organisms, but we're using DNA-based methods. And so if we do metagenomics, for instance, we always are measuring at a relative abundance level. We're measuring proportions. And sometimes you can say, well, this went up and this went down, but in the actual overall numbers, you'll have the opposite happening. So I don't have an answer, but I do have that statement.
Can I say something? Yeah, actually, there's a lot of tricks for the analysis of the communities. One is experimentally. So just like Jordan mentioned about PCR, because anywhere, because when you use PCR, you bias a lot. So actually, we did a few years ago, we did publish in UCMJ. So the same DNA, if you sequence 3 times, so you expect 100% overlap. So in reality, we get about 15%. So you get 80% of diversity. I think the field is equal. Most people ignore this part. So then you see we did another publication, M-Bio. So we did a mathematical modeling. We just assume the community no differences. So mathematically we can get 80% diversity without changing anything. Just change the depth of sequencing. So yeah, means most people ignore this part. I think this is a real issue if you want just diversity. Another issue about analysis. So if you use UCHIME or UPath or UCLUST, so the number of sequence OT you get 10 times differences. Remember, the program written by the same author. The same author wrote these 2 programs. If you use these 2 different programs, you analyze data 10 times differences. So I was involved in bigger collaboration with DOE Enigma. The same data, we use 3 lab processes, quite different in terms of OT call. It's the same pipeline. It's a lot of difficulty. I think the case that is so that's why people, we don't do absolute quantification, we don't do absolute comparison. All people focus on relative comparison because there's a reason there. Why should I do relative abundance, relative comparison? So even for the same sample, you use the same amount of DNA, you get sequence number maybe 10 times to 100 times differences. The same. No matter how do you control this? You get the sequence numbers 10 to 100 times differences. So that's why people just we do rare fraction analysis. We fix to like 200, like 120 or like this. There's lot of because I don't think anybody can solve the problem because this is the technology problem per se. So I think really people should be very careful, maybe how to analyze data, how to interpret the data. This is the key. How to design the experiment. I think maybe the committee should recommend how to really design the experiment, give the conditions that the technique can bottleneck. We cannot solve. I don't think anybody can solve right now. So how do we use the data more wisely?
Joe, it's a very important question, and I think it's a good place for us to take a break because definitely moving forward, it's something we have to address. So they say light snacks are provided. We are going to break now until 3:50.